Okay, we're going to continue this theme of evolvability. Can we have Goja Weh up here also from IMO? And she's going to discuss modeling selection for evolvability in the evolution of cancer therapy resistance.
Thanks, Sandy. Okay, now I need to figure out how it works. But hi, I'm Gosia. And I'm going to talk about my project that I started maybe a year and a half ago. But it's all based on the motivation that in multiple solid tumors that are treated with targeted therapies, we know that there's a population of persister cells that can adapt and evolve resistance. Now, evolving resistance is an evolutionary process, so it's based on diversification and selection. And because of that, evolvability, a trait of inducing heritable phenotypic variation that was just wonderfully introduced by Jill, thanks Jill, is going to be playing a very, very big role. However, I'm going to talk about evolvability from a completely different perspective. In fact, I'm going to be talking about mutator phenotype and how it affects this adaptation to treatment. So, mutator phenotype is essentially a tendency of cells to acquire more mutations over a certain time unit. And this tendency is also heritable. And what we know is that it can be caused by genetic and non-genetic factors. And also it's because it produces a lot of phenotypic diversity and genomic instability, it is also a hallmark of cancer. So how does mutator phenotype impact adaptation on treatment? That's a great question. But we do not know the answer to that question yet. And it gets very quickly very complicated. Because if we think about mutations, well, mutations can have a very different impact on cell fitness. What we know now is approximately 99% of mutations are neutral. However, there are some mutations that can cause population expansion and population extinction. So keeping that in mind and keeping how mutator phenotype can affect adaptation to treatment, we want to construct a mathematical model that will help us assess that question. So this model is an Asian-based model, surprise, surprise. And in this Asian-based model, we have a cell that can die with certain probability that is time dependent. More about this later. It can mutate with a frequency that is constant over time. And finally, it can also divide with a time-dependent probability. How time-dependent probabilities are actually executed, that depends on the mutation impact. So every time a cell mutates, we determine the impact of a mutation on cell fitness by withdrawing a random number X and comparing it to this distribution. And so essentially, if my number X is falling into the red region, the mutation is going to have a negative impact on cell fitness. If it's going to be falling into the green region, it's going to have a positive impact, and otherwise it's going to be neutral. Neutral is going to be happening 99% of time. And treatment in my model is modeled statostatic. And so essentially we just start with a population of persister cells that, using the whole mutation thing, they can increase the fitness of the population over time. And I had a beautiful slide to demonstrate how it works, but the slide doesn't work. But imagine that we start with a small circular tumor, and then over time, I have a tumor that is very orange and yellow. Anyhow, using this particular framework, we can now investigate what are the consequences of mutations interrogating cell fitness. And now imagine this as a pointer, and I'm pointing now to the x-axis, which is time, y-axis, which is number of cells, and then on the right side, we have different colors. The blue one is the fastest mutation rate, red one is the lowest mutation rate. And then this yellowish color is no mutations at all. And what we see is that cells without any mutations at all, they can only proliferate a little bit. They stay approximately under 30% of carrying capacity. So by 30% of the domain. And however, as soon as we get any mutation, or like as soon as we have probability of mutations, cells can actually accelerate adaptation to treatment, and they can achieve carrying capacity at some point in time. How fast that happens depends on the mutation rate. And so we see that cells with highest mutation rates, the blue ones, they do it fastest. So we can conclude that mutator phenotype accelerates adaptation to treatment. Well, despite that, if we look at the cell fitness here is defined as the probability of division minus probability of death. Well, we see that cells with highest mutation rates are actually having the lowest fitness over a long time. And in fact, if we look on the right side at these box plots that are taken from the very last time point, well, we can see that these the population with intermediate mutation rates have the highest fitness. Interestingly enough, if we look at the variation of cell fitness across cell populations, cells with highest mutation rates have actually the highest variance. But what is also interesting is that despite them having this huge ability to mutate fast and a lot of times, they stabilize their variance very quickly. So I am not fully understanding it. If you have any idea of why this is happening, let me know. Nevertheless, we've got some characteristics about how mutator phenotype, or the ability of having a lot of mutations and inducing them over time, accelerates adaptation to treatment. But we do not know exactly the dynamics of how it happens. So what I'm going to do here is that for all of my replicates, I'm going to look at the time point where we observe the most rapid population growth. And then I'm going to arrange my data such that everything is centered about individual replicates' time of maximal cell proliferation, or maximal population growth, sorry. It looks like this on the slides. And again, number of cells on y-axis, time relative to the maximal population growth on x-axis, and then each different plots is different mutation rates. So what we observe here is that cells, as we move towards the right, as we are increasing mutation rate, the adaptation is very rapid, and it's also not having a lot of stochasticity. It's almost like a it's almost like a step-like behavior. But as we're reducing mutation rate, it's actually getting more stochastic, it gets more uncertain, it gets much more gradual. And if we look with that and compare it to the cell fitness, well, we observe that cell fitness needs to increase prior to the rapid population expansion, but how rapid it happens depends on the mutation rate as well. So I think this is really cool. I'm not sure I fully understand that yet. Finally, very quickly here, mutations, both positive and negative, accumulate. But what we see is that cells with higher mutation rates accumulate a lot more positive and negative mutations than cells with lower mutation rates. And okay, I know I've been talking a lot. So far, the conclusion from all of this I've been talking about is that yes, persister cells, they have approximately zero net growth and they can adapt to overcome the treatment. However, the zero net growth conditions can technically be changed because we might change treatment. Cells can also be put into different environments. So what if our cells are kind of facing the extinction, or what if they are doomed? And would mutator phenotype provide any benefit for these populations? The answer is yes, but it's not the only factor. So I wish I had more time because this is one of my favorite parts of this work, but whatever. So cells that are potentially facing extinction, they can adapt two strategies to have, or they can have two strategies to overcome possible potential doom. One of them is to be lucky, simply by having very high advantageous fitness from the very beginning. And the other stuff, or the other strategies that cells can have, is the evolutionary rescue provided by mutator phenotype. Yes, so this is, so essentially what it means is that cells can have, cells that are mutating a lot can accidentally acquire enough of positive mutations to allow them to survive. And there's much more detail into that. Again, feel free to ask me later on. But I kind of want to end up here and leave you with this particular question, which is, sure, we have now learned that cells having high mutation rates, they can adapt to overcome treatment very fast, and they can overcome potential threats. But they do have a very very variable fitness, and they have lower fitness than cells with lower mutation rates. And the question is, if this is a high enough price for cell populations to, to be there and potentially being able to survive long term when challenged or when competing with other cell populations. Is the ability of having a lot of mutations and producing a lot of mutations, is this beneficial under all circumstances? We do not know, because all of this is actually depending on the context. And this is how I would like to actually encourage all of you and myself to think about the trade-offs and costs of resistance as something that depends on the context. And last but not least, we want to actually validate all of this experimentally. We want to take cells, allow them to produce a lot of mutations, and then count, see, compare cells with different mutation rates. And here I'm gonna stop now. This is my cat, she's lovely. And I also want to say thank you to my wonderful advisors, David and Andre, and everybody from my group, everybody from the IMO, and my cat again. So thank you.
Really exciting stuff. So I might have missed this, but in your case, as your cells divide, they accumulate mutations. They keep doing this as they mutate and they divide, and it keeps happening? Or is it like, oh, you mutated and you stop mutating now?
Yes, cells keep accumulating mutations, and the probability of mutations that stays constant over time. So,
okay.
Yes.
So then in that case, it sort of feels like this gambler's ruin problem, right? Where if I'm gaining mutations really fast, then it's okay as long as I'm getting positive mutations. But as soon as I get one deleterious mutation, I'm out. And so if I keep running the simulation long enough, I'm always going to go out. Mm-hmm. Right? And so your population will automatically drop. So fast mutations in general, if you run it long enough, will end up being really bad. Is that?
I mean, yes. However, it again depends on the context, right? Because sure, we can let things run for a very long time, but imagine that given that mutations can actually help cells to sorry, again, I'm very jet lagged. So if we subject our cells that we know now that they can adapt to treatment very fast, if we subject them to a variable environment, yes, they will be having a fitness disadvantage over a long time because they keep accumulating mutations, but it potentially will help them to adapt to treatment. So I don't know actually, because I think everything here really depends on the context, and that's what I want to do next. Essentially, look how different context, how having mutator phenotype will help cells in different scenarios.
Cool. Thanks.
Mhm.
Hi. Very interesting stuff. I'm wondering about the way you think about it. So especially in the last experiment, you give agents to increase the mutation rate if you want to test it experimentally, and then see how they adapt. And that's also how your models are set up. But in order to get a mutator phenotype, that has to evolve first. And that can maybe involve the loss of a tumor suppressor gene, which requires 2 mutations. So it's going to take, it's an extra step. So how does it, if you take that into account, how would that influence sort of your arguments? Because you could be potentially faster if you just skip the genetic instability and you grow to a large size. And once you're like 10 to the 10 to 10 to the 12 cells, you generate lots of mutants anyways, right? So there's some sort of a trade-off going on there.
Yeah, that is a great question. So let me think. So in the experiments that we are planning to do, we want to subject cells to different mutagens. And there are different ways of doing that. And I could imagine that how we will be doing, how we will allow cells to generate a lot of mutations over time will be having a lot of impact, because if we say if we are doing this with ApoBec, which is an enzyme that allows cells to produce a lot of mutations, well, it first needs to be expressed. But if we say put a little bit of UV to increase to do DNA damage, well, there will be. I could imagine these influencing the results too. Now. Sorry, what was the rest of the question?
Well, so what you're looking at is instability, right? Genetic instability, essentially.
Yes.
And the question was like, if you have an extra step that you need to produce that instability, will that change your conclusions? Because it's going to slow down the whole process, because you first have to evolve genetic instability, and during that time, a tumor could stably grow to large numbers, right?
Gotcha. Okay, so of course. The answer is, of course, it will be slowing down the process. However, I do not know if you noticed, but my timescales and everything here is actually fairly variable. The thing is that what we want to do here is to create a model that will potentially demonstrate it from the theoretical point of view. We do not aim to precisely validate this. We are looking for more of a qualitative confirmation of our hypothesis. So yes, timescales will be varying, but I need to figure that out when we actually get the experiments done. So
all right, thank you.
Hi, Gosha. So
hello.
Great talk. It came to my mind right now. I didn't have a chance to ask in the past, just when you gave the talk at Moffitt, but just I'm curious what you think about different type of drugs also introducing evolvability to the cells. Do you have some data or model on this? I'm asking, by the way, this just because of this particular thing, just to sum up the whole thought. For example, you use alectinib for the example, and in lung cancers, there are other targeted therapies, right? EGFR inhibitors or so. And after those therapies, it's known that the tumor relapse occurs, but actually it is also known that different targeted therapy options cause very different sometimes like overall response rates, or let's say, relapse occurs at very different periods. So that might mean actually drug type also might. And I'm just curious if you have any ideas or data.
So the short answer to your question is I have zero idea, because we haven't tried that yet. However, keep in mind that in real life situation, when when we actually will be looking for that from the in vivo or in vitro perspective, with we will be, not all cells will be having the same mutation rate. They will be having, some cells might be some cells might be mutator, having mutator phenotype, some may not. So there will be also a component of cell competition for space and resources and everything. And I could imagine that if that treatment will be having quite a lot of impact. And given that our tumor populations are already very heterogeneous prior to treatment, there will be selection going on, and there will be quite a lot of again, there will be competition, there will be population expansion, there will be a lot of stuff going on. So I do not know. I'm curious to see that in the future, because this is what we plan to do at some point.
Yeah, sounds good. Thank you.
Thank our speaker one last time.